question session. Professor Yang?
Thank you, Dr. Hu. I have two questions. One is about the antibody, the vaccine program. You mentioned about 10 to 20% of the patient eventually has developed immunity by affinity maturation. So has anybody analyzed the antibody pools in those patients and find out what kind of the structure or particular combination that are important? Yeah. And the second question is for the GSK for the prevention drug. Have you tried to add it into the cocktail so that you will make the cocktail therapy more effective? Thank you.
Okay. There's been enormous effort put into analyzing the structure of those antibodies, and even some of the structures of the antibodies, the somatic variants of those antibodies. So at the atomic level, we understand a lot. And sequence-wise, we know how the antibody genes evolved to get there. But the problem is, while we understand the process, we don't know how to induce it. And that's what the vaccine needs to do. The other problem is that, you know, we have certain antibody gene sequences. And if you look at the those are called the germline sequences before the affinity maturation occurs. Those germline sequences, if you make those antibodies, they don't bind to the antigen. Or most of them don't. So how do you induce that first step is not clear yet. The subsequent steps are clear because once you get some binding, they bind better and better and better with time, with affinity maturation. But if the germline sequence doesn't produce an antibody that actually binds, or it binds at an affinity that you can't even measure, that's part of the problem. And then it takes so long for that to occur. So if your vaccine regimen has to recapitulate that, it might take 4 or 5 years. That's not, you might as well go with the drugs, right? So that's part of the problem. The 744 second question, if you combined it with oral pills, then you'd be getting an injection and taking pills every day. So the ideal, this is what we and others are now thinking about, the ideal thing would be to change yet again the paradigm of treating HIV/AIDS from taking a pill every day to taking an injection every 3 months, let's say. But you need a cocktail of drugs that could be, each of which could be formulated for slow release. There's now a second drug called TMC-278, which is not an integrase inhibitor, but a reverse transcriptase inhibitor that could also be formulated for slow release, but it's every month. But now, given this prime example of 744, you could see people in the industry are thinking that it would be fantastic if we could shift the paradigm, at least for a lot of patients, to a quarterly injection, and they would take care of it. And the results would be even better because you don't have the adherence problem, as long as you get the patients to come back. And these days, there are all sorts of reminders to get the patient back, through messaging and other means. So the scientists are all thinking about it, 动脑筋, and I think it's only a matter of time. If you think about drug development, and I lived through this in the 1990s, a lot of protease inhibitors that were developed. So those are, in general, a powerful class of drugs, most of which were extremely aqueous insoluble. And in fact, you know, the chemists would call them bricks and then put them aside and not develop them because they would be bad candidates for oral pills, but they would be ideal candidates for slow release if they have several other potent, the properties like potency and slow metabolism. So everybody's going back to their pool of chemical compounds that have activity and re-examining it for another purpose. So it would not surprise me 5 years from now we would have regimens that could be given every quarter.
You showed a list of those variants you tried. How did you pick up those variants? Can you actually say, I use a computer to model those and then select some sort of promising ones and then try sort of wet lab experiments?
Right. Well, we try everything. And in fact, when we make I'll take even one step back. When we made the bispecific, we gave it a lot of thought about which pairing would make sense from the biology, from our understanding of the biology of virus entry, from our understanding of the structural aspect of receptor, co-receptor, and the envelope spike. But in the end, the best pairing was empirical. Now, in terms of what you just asked about the somatic variants, again, the final outcome was empirical. But we really tried to predict whether certain residues were more hydrophobic or hydrophilic, and whether they would affect it. And in fact, we work with someone who perhaps is a leading expert in the area named Bernhard Trout at MIT in the chemical engineering department. He's dealt with these issues a lot for industry. But the predictions are hard. But once you have a form that works, explaining it is easier. Rationalizing it is easier. But sort of starting from no knowledge or bare knowledge, it simply was not possible to predict a priori what will come out. So it's been pretty much empiricism.
Yeah.
Please accept my apology if I ask something you have mentioned before. I just don't want to misunderstand anything. So can you tell us when and where the clinical trial will be taking place, and will Hong Kong gay men include in the trial? And my second question is how many years we need to wait until the protection drug can be used among the high-risk people all over the world?
Yeah.
Thank you.
Well, I could tell you what I know. What I know is that we're already doing the phase 2 study. All the subjects in our phase 2 study have enrolled already, and the follow-up is less than a year. So by spring of next year, most of the data would come out from the phase 2 study. So that we know quite confidently. What I cannot answer you is when phase 3 will start, where it will be conducted, because that's not my decision alone. It's we have many partners involved, not just the company, but potentially lots of investigators. It's going to be a multinational, multi-center study, and it's a complex process which I'm trying to manage with help from some colleagues like Chen Ziwei in the audience. And exactly where, we're going to pick locations where infection rate is high. That for sure. And we're going to pick, and that's because I said during my talk, if the infection rate is high, then your cohort size is small, and your follow-up duration could be shorter, and your cost is lower. So most likely it's going to take a year or a year and a half. Exactly how many? It will be probably 500 to 1,000 subjects. Exactly which location, we can't say until we decide together with all the collaborators. Would Hong Kong be included? Well, Hong Kong would have to be factored into those considerations. You know, Hong Kong infection rate is 3-4 times lower, then it may not be as competitive as other places in our sort of overall considerations.
Okay. Yeah.
I have a question about the GSK drug, which you mentioned this integrase strength transfer inhibitors. My question is the mechanism of action matters, or is the long-acting effect matters? Because the reason I ask is that there are two existing FDA-approved drugs for integrase. Mm-hmm. So could you comment on whether those two FDA-approved drugs could be used as nanoparticles to formulate a drug which make faster for approval?
Right. Those, I think you're referring to two other integrase inhibitors. Actually, raltegravir from Merck, dolutegravir also from GSK. In fact, this is an analog of dolutegravir. 744 is an analog of dolutegravir. So, and those are approved, but they are not so aqueous and soluble. Their metabolism is faster, and their melting point is different. They are not as suitable for long-acting formulation. But your question is still a good one. Is it the drug or is it the slow? I think all drugs do work. If you look at the clinical trials that have been done using reverse transcriptase inhibitor and look at the subset of subjects who had high drug level, that's about 90% efficacious in human beings. So, and those are reverse transcriptase inhibitors, not integrase inhibitors. So in general, you know, HIV replication is a circular process, right? And if you want to cut that circle, essentially you could cut it at many different points, as long as you cut it completely. And that's how I see it. So I think in the future, and TMC-278 is a reverse transcriptase inhibitor that's now formulated for long acting. So there'll be more.
Can I follow up that question? Is it possible to just go back to find those drugs that the medicinal scientists originally thought it's not very good?
Yes, that's exactly what I'm saying.
So it's just
most of them that have been put aside because of insolubility could be pulled out for repurposed.
So the rule of 5 should be re-examined.
For this indication, yes.
Okay, thank you.
I wouldn't say for everything else, but for this purpose, exactly.
Yeah, I have seen that. Yeah. Yeah, please.
Good afternoon, Professor. That was a very, very interesting talk. I'm a veterinary pathologist. In the early 2000s, I was involved with a lot of research in HIV. Also noticed in the introduction, you were the 1996 Time Man of the Year. I want to go back to Time magazine. In 2009, they had the top 50 inventions, and the #1 invention was a AIDS vaccine. And at that time, scientists really couldn't tell the importance of this vaccine because if they gave 2 doses, it didn't decrease the individual's chance of attracting the virus, but it was like 33% decrease of them getting infected. I don't remember the exact stats, but I just would like to for you to give an update or give your opinion about that invention of the year. Thank you.
Yeah, well, those are picks made by journals and journal editors, not really by scientists. I think what you're referring to is a combination of 2 different vaccines that have been tested in Thailand. Based on your description, I surmise that it is the result of what we call RV144. It's a study in Thailand and show 2 vaccines. There are 2 different vaccines. One you give multiple times, and then you come and give the second vaccine multiple times. I think a total of 8 injections over a course of almost a year. And it showed a range of protective efficacy of 26 to 31%. Now, you know that when many of you do experiments, when you see such a small effect, you have lots of questions. And so the field is essentially waiting for that trial to be repeated in a different population and see if, I mean, I think most of us take it at face value. There is something there, but it's so little. A vaccine that's only that efficacious will never be advanced. It generally has to be 50+%, I think. But it becomes a tool to say what's in that vaccine that's mediating protection, and there are lots of studies that have taken place subsequent to that report. But it is certainly should not have been a breakthrough of the year, in that it's not even applied to anybody yet. But it was clearly viewed as advance in the field. It's a positive step in the right direction, but it still requires a whole staircase to take you to where you want to be.
Okay. Other question? Oh, Professor Chen.
I just want to know whether there's still any follow-up on antigen presentation and good old-fashioned immunization.
Yes. My group received a grant from the Gates Foundation prior to this work to do exactly that, to focus on the antigen presentation features of a dendritic cell and how to activate, how to get the antigen to the right cell, how to turn on that cell. So we were funded to do sort of targeting immunization approaches and adjuvant approaches that would stimulate the dendritic cell. And in fact, we have separately, which I don't talk about, we have a glycolipid that's mechanism-specific adjuvant. It's a CD1D binder that turns on dendritic cells and via NKT cells. And that's, we actually are taking that product into clinical development very soon. The GMP product is already made. So that part, the adjuvant part, would work very well, but the targeting strategies never materialize. Fundamentally is we don't know how to shape the immunogen. We could get it to the right place, but how to shape the immunogen so that it would consistently induce the neutralizing antibody part. That part is still missing, and so we don't quite have it. But sort of the spin-off benefit of all this intensive study of HIV is, I think, it's going to benefit vaccine development in general, even if we don't come up with one for HIV. All of us who have spent now years working on HIV, sort of hitting our head against the wall for 2 decades. When we look at another virus, we think it's really easy. If you look at Ebola, we could do that, no problem. When we saw 2003 SARS, Zhiwei made one SARS vaccine, it protected. It's easy. But HIV is a real challenge. And if you think about all this, how an antibody response occurs to a pathogen like HIV, that's never been done before. If you think about it, all these dozens of antibodies, the structures are solved, exactly how they bind to the virus is known. How then the gene sequences evolved, and what % of the antibody repertoire is directed to the virus. All these things are worked out for HIV in great detail. And that's never been done for anything else that I know about. And that's really remarkable science, but the fundamental problem is we don't know how to take that knowledge and turn it into a vaccine. Not yet. It seems that another critical breakthrough in another sector is required. And I think the problem is so challenging that perhaps it will be passed to the next generation.
So, may I have a question from next generation? Yeah. Okay.
Thank you very much, Dr. Ho, for your fascinating lecture. This question is somewhat like the former question that you mentioned in the short foreseeable future that we may not have an effective vaccine towards HIV infection. So since you are the person that's standing on the cutting edge of this area, I want to ask, in your opinion, can we eventually have a cure to AIDS disease or an effective vaccine towards HIV infection? And if the answer is yes, in your opinion, how long do we have to wait for that? Thank you.
Yeah. The toughest questions are ones looking forward, right? Where are we going to do the trial? I don't know. When are we going to have a cure? I don't know. When are we going to have a vaccine? I don't know. I think, just on a more serious note, I think the cure is equally challenging, right? As a vaccine for HIV. We have a virus that integrates into the host cell, and the host cell is a memory CD4 T cell. And you know, memory cells, immunological memory could be lifelong, and hence the virus could be lifelong. And we don't have, and we know now when you control a patient very well with combination antiretroviral drugs, we know the virus is still around. It's not gone. That's why we don't have a cure. As soon as you stop the drug, the virus will rebound within weeks and then start to do damage again. So we know we can't stop. And when you look at the state when the virus is well suppressed, basically it's about 1 in a million mononuclear cells in the blood that carries the virus, infectious form of the provirus. So it's a small percentage, tiny percentage. So how do you activate? And it's silent, so it's latent. So it expresses no viral protein. So it's indistinguishable from the normal memory CD4 T cell. So how do you specifically turn that cell on? So the drug could, so that that cell would die and the drug would prevent further infection. It's very hard. And we don't have very sharp tools to turn on cells, right? We have very blunt tools to turn on our T cells. But when you turn on your T cells or your immunological cells, only a few percentage, you have a shock-like syndrome. So you have a barrier there. And you could do the math. Those of us who went to Caltech could do this math very quickly. That if it's 1 in a million, and you could only activate a certain percentage, how many activation events you need, it would exceed the lifetime of that person. So it's impractical, and every time you do it, you're afraid of killing the patient, when the patient is now doing very well. So the balance of the equation is that you cannot activate non-specifically, because you are more likely to do harm than good. And so, and I worked on cure from 1996 through about 2002. And then I said, enough, because we were not getting anywhere. And it's like shooting for the moon. You're going for the moon, but you're back in the days of Galileo. Yeah, it's possible someday, but it's not possible back in the days of Galileo until you have all the other breakthroughs. And this is why I'm really upset by some of the statements put out by global leaders that created an impression that AIDS is under control. And these slogans really bother me. You know, towards zero, or getting to zero by 2030. It's not going to happen. This epidemic is going to be with me for the rest of my lifetime, and unfortunately, I think for the rest of your lifetime. It's not going to go away that fast. It's still spreading, 2 million plus cases per year. So I think that problem is extremely, extremely difficult. And I think we all have to know our own limitations. Yes, everybody would like to cure cancer, but that knowledge accumulates in a stepwise fashion. And if you just want to, it's like a 5-year-old trying to hit a home run against a pitcher who throws. Well, baseball is a bad example for Hong Kong, but it's just not going to happen.
Yeah.
Thank you, David Ho. Yeah, it's a very amazing lecture. Yeah, because I am also working with bispecific antibodies, so I am very interested in that part. And I'm not sure if you have found that sometimes a bispecific antibody is working even worse than the parental antibodies.
Yes. Yes. I said you see all sorts of patterns. For majority of the antibodies, they're about the same, but there are some antibodies that are worse. And it makes sense because bispecific antibodies, you have the one arm affinity, the other arm affinity, but you don't have the two arm avidity.
Yeah.
And for certain situations where avidity plays a very important role, it's going to affect your outcome.
Yeah.
So
yeah, I think it's a very tremendous work. So is there any implications for the parental antibody selections and the combinations, not just for HIV, maybe for cancer therapy?
Well, I don't know if we could directly take what we have learned from HIV and apply it to specific cancer therapies using bispecific antibodies. I think one lesson I've learned that may be applicable is a lot of this is empirical work. And we always think that we're very smart, we know a lot, and therefore our thinking will, and I've found that while, there are many smart people and who are very logical, what we do know is actually dwarfed by what we don't know. And when you look from that pinhole, your perspective is very skewed. And so while it makes a lot of sense to be rationally designing things, and that should be done in areas where knowledge is extensive, but where knowledge is lacking, and what we know is actually such a small part of what we know in total, or what we don't know. It just doesn't work very well. So I think it unfortunately comes down to trial and error or empiricism.
Just keep trying.
Yeah.
So thank you very much, Professor Ho. So thank you very much for the audience for your active participation. So thank you.